Okay, good morning, everybody. Welcome to this poster session on genetic resources and crop improvement. In this session, we will have 4 posters and we'll go from wild relatives over phenotypic diversity, genetic diversity, up to value chain creation and usefulness of neglected crop species. So now I would like to introduce the first poster, which will be presented by Martin Wiele and will be about the origin of datepine. Mr. Wiele, the floor is yours.
Can you see the slides?
Yes.
Just a moment from my side. Yeah, good morning from my side. My name is Martin Wiele, as mentioned, and I will introduce you today the origin of date palm, methods to distinguish between putatively wild and domesticated species. Just a moment. Sorry. Yeah, so date palm is a widely recognized keystone species of arid and semi-arid ecosystems across North Africa, Cape Horn, the Middle East, as well as the Indus Valley. It belongs to the few perennial and intensively domesticated species whose wild relatives and putative centers of domestication are not yet recognized, although actually many researchers assume a Middle East distribution of wild date palm. In recent years, studies increasingly suggested a North African center of origin, as well as rarely a center in the Maghreb region around Mauritania and Morocco. According to FAO, date palm accounts for roughly 1% of the global fruit production and is therefore an important agricultural commodity. Due to the unclear evolutionary and domestication processes and its agricultural importance, research in recent years has gained momentum to search for its wild relatives. It's driven first by the need to increase the dynamics, increase the genetic pool for necessary selection of adaptive management to mitigate climate change, and second, scientific interest in the likely complex evolutionary process of this species. What we did in this study is a multidisciplinary approach by reviewing current trends in date palm research. We use archaeobotanical, such as phytoliths or pollen analysis, as well as archaeological records, such as cave paintings and reliefs from historical buildings, to unravel date palm's ancient occurrence and use over space and time. We look at varietal diversity per country by following the Vavillov's center of origin theory. We further consider climatic conditions between 130,000 years before Christ and today by applying species niche modeling with coordinates of putatively wild accessions of date palm. Lastly, we synthesize literature and own data to better predict putative regions of wild date palm. I present here now 2 outcomes of this review of this study, and the first one is this Vavilov center theory, where the number of date palm varieties per country revealed a more or less balanced distribution with a clear focus on Northwest Africa, Algiers region. When looking at the cultivated areas of this region, varietal richness was concentrated around Kuwait in the United Arab Emirates, but also a higher share in Mauritania. Species niche modeling reveals 3 centers of date palm origin across all available climate periods that are Morocco, so also again Northwest African regions I present here, the Levant around Egypt, Israel, and the Persian Gulf coastal regions. Despite many methods used and studies known, potential centers of date palm origin remain obscure. We tentatively identified likely regions of putatively wild accessions supporting the theory of 3 domestication centers. This indicated also the necessity to reconsider regions with low accessibility, such as the Arabia Deserta. Furthermore, the study underlines the need for a more structured and collaborative research program and offers potential approaches to study other domesticated species of interest. Thank you very much for listening to the talk.
Okay, thank you very much, Mr. Wiele, for this nice and informative presentation. Now I wonder whether those who are in our Zoom room have any direct questions to the presenter. If not, I might start. So I'm also, as a plant breeder, very interested in wild relatives, but I wonder the advantages and disadvantages of using wild relatives in the genetic improvement of date palm. Would you have an answer to this question?
Can you please repeat the question?
I'm a plant breeder by training, so I'm interested about the advantages and disadvantages of using wild relatives in the genetic improvement of date palm.
Yeah, certainly there are from the wild date palms that are not yet known, and it's even not known if there are any wild date palms left, would have the potential of higher resistance for diseases, but also maybe drought, more drought resistance. So this could be an approach for future breeding, because we see that also the domesticated, highly domesticated varieties available are genetically very narrowed and are susceptible to many stresses, environmental stresses.
Okay, I see in the chat you are asked to turn off your screen sharing.
Yeah, wait. Yeah, no. I
Wow,
also looking for the screens for the people, yes.
But no problem. Questions from the audience? I in the chat so far. Maybe just to summarize the various uses of date palm.
Yes, it's a part of its fruit, which is of course the major commodity. Also, the wood can be used for construction purposes. Leaves are used for thatching roofs. Some people produce baskets, so it's also locally produced products. Actually, yeah, sustainable products that are also degradable again. Also, there are approaches that, for instance, the seeds are pressed, and it can be a feed additive for livestock. The fronds or the leaves are also eaten by cow, for instance, or particular cow are quite for many palm species. They also use the fronds as additional feed. These are the main. And of course, it has a great recreational value these days. If you look at Middle East or North African as well as Southeast Asian regions, date palms are central elements of city planners. So providing shade, beauty, simply beauty is a perfect or important aspect.
Yeah, it's really a multi-purpose crop, which can also really contribute to smallholder farmers, I believe.
Mm-hmm.
Nutritious crop, also high energy value. Yeah, lots of uses. Very exciting crop. I'm happy to have a session. And I think now we could move to the second poster presented by Claudio, right? On the phenotypic plasticity of roots of Cocomia culiata in Western Minas Gerais, Brazil.
Yeah.
Claudio, please start sharing.
Yes. Can you see it?
Yes.
Okay. So, my name is Claudio. I'm forest engineering student here in Brazil, and I'm going to explain the phenotypic plasticity of roots of a cocomia culiata in Western Minas Gerais, Brazil. So, endemic to the Americas, A cocomia is an oilseed palm tree with a high oil productive and agricultural advantage compared to Elysis guineensis. Its fruits have four layers: a protector exocarp, a flesh mesocarp, a hard endocarp, and an endosperm. This study aimed to understand if the orange of the genotype have impact on the fruit's biometrics. So in the harvest season, two different ecotypes were selected. Ecotype from the Macaúba Active Germplasm Bank of the Universidade Federal de Viçosa in Araponga, Minas Gerais. Were selected two ecotypes, ecotype D and ecotype 8. Dry and fresh weight of all four layers were collected, uh, also thickness from the exocarp, mesocarp, and endocarp, and a higher diameter of the endocarp. We saw that the fruits show a natural ecotype difference. For example, fruits from ecotype 8 tend to be bigger with a harder endocarp, and both ecotypes show a similar size proportion of each layer. And in both ecotypes, the endosperm size, dry weight, dry matter content, and density are comparable in contrast to mesocarp. However, ecotype 8 show a similar, uh, show a low dry weight and a dry matter content. We can also see that more mesocarp can be gained from ecotype D, suggesting also a high amount of oil which could be extracted. Therefore, fruits show high plasticity, increasing the phenotypic characteristic variability, which is often important for developing a cocomia into a crop through breeding. Because of COVID-19 pandemic, oil extraction from the mesocarp and the exocarp couldn't be done, but an assessment of more ecotypes from other regions of Brazil and South America. But it will be done in the upcoming months now that everything is going back to normal. And thank you for listening and looking forward to any questions.
Thank you very much, Claudio. This is also a very exciting plant species to work on and to look at. Are there any questions from the audience here in the Zoom room? I don't see any questions on our question and answer side. And maybe to start off the discussion, how you are defining phenotypic plasticity, because to me this also has maybe a different meaning. So how would you define phenotypic plasticity?
Well, I would define phenotypic plasticity as a capacity of the fruit to adapt his natural characteristics to a new environment.
So you mean adaptive phenotypic plasticity?
Yes.
Which I also know, and I agree that is a very important character, which could contribute also to yield stability. But I wonder whether in your case you really assessed adaptive phenotypic plasticity or whether you just assessed phenotypic diversity of these ecotypes.
Sorry, I couldn't hear well the end of the question.
Yes, I was wondering whether given your experimental design, did you really investigate adaptive phenotypic plasticity, or did you rather describe phenotypic diversity among those ecotypes?
Well, I would say that it's it's not a diversity, but it's not like a phenotypic diversity, but I would say like yeah, have a phenotypic necessity in the fruits. Not are diverse because like we have fruits from many regions in South America. So each one have a natural ecotype characteristics. So we want to see this variability.
Okay, well, maybe we need to look a little bit deeper into this. Are there other questions from the audience? Yes, Mr. Wiele?
Yes, Claudio, thanks for this talk. I'm interested in the exploration status in Brazil or in the region of occurrence. Like, how much is it commercialized? Are there plantations, or is it just maybe on a small scale home garden level, or what is the status in Brazil?
Well, it's not too much commercialized. We want to see like the cocomia culiata with the Elysis kinesis, which is very commercial here in Brazil, and we want to introduce it like in the market to the industries to see that it's like it has a high oil production and agriculture advantage. So we want to introduce it now in the market.
Mm-hmm. Okay. The question to your methodological approach, maybe I didn't get it from the slides, how much you sampled and from where, like how large was the range of sampling location?
Oh, yeah. We collect in the harvest season. We collected like 15 fruits of each plant, and it was like I don't recall right now in but it was like 7 or 8 plants of each ecotype.
Mm-hmm. So rather still a preliminary assessment of diversity, because normally you would certainly need a larger extent of.
Yeah. We want to go deeper, but because of the COVID pandemic, we couldn't.
Mm. Okay, thank you.
Okay, yeah, thank you for the contribution to. I think with those neglected, it's also neglected and underutilized. Those value chains is really a promising approach in order those genetic resources. If there are no other questions, then we could move to the next presentation from II Tant.
Yes.
Genomic studies of Myanmar rice varieties using DART and SNP markers. So you said you would show the video, is that right?
Yes, it's right.
Mm-hmm. Please give a sign in the chat when to start the video. You don't hear the tone right now. We don't hear the tone.
Hi everyone, my name is Ethan. I'm a PhD student
Start.
from COLS in Prague. Today I am presenting genomic studies of Myanmar rice variety using DRDS net
Right
markers.
now.
As you all know, local
We
rice
don't
varieties
hear.
are well adapted to local growing conditions and are valuable genetic resources for further improvement. Therefore, this studies was carried out.
So can you hear? I don't hear anything.
No, I don't hear.
Yeah, we don't hear.
Technical difficulties. But our technician is working on it and supporting us.
And we have still sufficient time, which is good.
Yes.
Yes. Hi everyone, my name is Ethan. I am a PhD student from COLS in Prague. Today I am presenting genomic studies of Myanmar rice variety using DRD SNP markers.
Second. Sound is good, but the picture is not good. Still technical difficulties, but hopefully they will be solved soon, because we now heard the sound, but now only the image of the video is not very clear. It's cut off actually. Was almost there.
Hi everyone, my name is
We heard the sound. Yes, now the picture is good. Oh, now it cut off again. Sorry, I'm really sure you were so well prepared. Yeah, now it's visible.
Hi everyone, my name is Ethan. I am a PhD student from COLS in Prague. Today I am presenting genomic studies of Myanmar rice variety using DRD SNP markers. As you all know, local rice varieties are well adapted to local growing conditions and are valuable genetic resources for further improvement. Therefore, this studies was carried out to identify genetic diversity of Myanmar rice genotypes using diversity array technology. DRD markers enable whole genome profiling of crops in the absence of sequence information. It has generated two types of markers, SNP and silico DRD. Before genomic analysis, we check first the quality of markers using quality control parameters such as reproducibility, core rate, and PIC. In Figure 2a, 99% of total markers in both SNP and silico DRD showed greater than 95% reproducibility. In Figure 2b, 83% of SNP markers had a core rate greater than or equal 75%. In silico DRD, 58% of total markers had a core rate 85 to 95%. According to polymorphism information content, 41% of SNP and 49% of silico DRD were observed in PIC class between greater than 0.3 to less than and equal 0.45. So, based on this marker quality, they were determined to be highly informative markers for genomic analysis. We studied with population structure. The optimate K value indicated that two set populations. And Figure 5 shows the proportion of membership of individuals in each cluster. The mobile result revealed that high FST indicates significance in differentiation populations, and low NN values means low genetic exchange among that population, which leads to maximum diversity among population level, which is 74%. And that table 2 provided genetic diversity index, and population 2 had the higher level of genetic diversity than that of population 1 based on Shannon Information Index, Diversity Index, and Unbiased Diversity Index. To conclude our study, DRD platforms can generate highly informative markers for genomic analysis in rice. Based on our findings, the rice panel was genetically diverse, and the level of diversity could be the basis for developing new rice varieties. Thank you very much for your kind attention.
Thank you very much. I for this very well prepared presentation.
Yes, thank you.
Are there any questions from the direct audience in the room? I don't see a question on our question and answer panel. Maybe I could start off the question. Could you just describe for the non-geneticist the advantages and disadvantages of DART and SNP markers in characterizing such genetic diversity?
Yes, and that is and actually the diversity of technology has included both the DRD and silico DRD and SNP. They can generate both types of markers, silico DRD and SNP markers. So actually, this is the combination of the next generation sequencing. It is combined with the next generation sequencing method, but it is the what we can say is a highly throughput, and it can generate thousands of polymorphic loci in a matter of hours, and also it doesn't cost much, and we can do a lot of we can do a lot of varieties. I mean, a lot of genotypes in a short period of time. So this is the advantages of the DRD-seq technology.
Okay, thank you. Are there further questions from one of you? So based on, oh yes.
Yes, the question, these traditional varieties you observe, they are land races or they are defined varieties or cultivars?
And we used 117 genotypes, and only 5 genotypes are the improved varieties. That we included that one because we want to make the comparison between the land races and the improved rice variety. So 112 was the land races and 5 was the improved varieties.
And they were from different villages, I guess, or from different farmers?
Yes, but in the one region, in different villages.
Okay.
And based on your results, what are your recommendations to rice breeders?
Yes, actually, this sequencing the genome, Myanmar rice land races is, as far as I know, it is the first report in our country especially for the Myanmar traditional rice variety. So we can do the further studies such as exploring the phenotypic traits which are collected with the in each individual signs. So we can do the marker assistant selection and the genomic genome-wide association study by using the sequencing data of the land races, which is my findings from my research. So we can make, we can make the field experiment to explore the qualitative data to compare which traits are connected with which chromosome. I mean.
That was exactly my next question. Diversity relate to the phenotypic diversity of those varieties.
Yeah, actually, I did already phenotypic diversity, but I did only one season. So we confirm it cannot be concrete information. We can do it another season, 2 or 3 seasons, and then we can do it.
Yeah, it's a good start, one season, but
yeah,
for sound findings.
Yeah.
Okay, are there any further questions? That was, if not, I think we could move to the poster presented by Anna Manurova. Zambian neglected species, oils and cakes, composition of traditional oil-bearing trees. Yeah, okay. So I think we are.
So can we start the video or? Dear audience, my name is Anna Maňourová, and together with my colleagues, I would like to present you our project targeting Zambian neglected species, in particular oils and cakes composition of traditional oil-bearing trees. The traditional food production systems, together with wild plants, local crops and landraces, are recently quickly disappearing under the pressure of agriculture intensification. The rediscovery and support of the indigenous neglected plants should be therefore of high priority. In western province of Zambia, most of the traditional crops were substituted by maize, mango and cashew nut. Having only limited access to nutritional and well-balanced food, local communities are threatened by malnutrition. Therefore, our study focused on the chemical composition of oils and cakes of 3 traditional Zambian oil species which are commonly utilized by local communities. From the 2 graphs on the left, you can see that the oils of Parinaricura telefolia and Schizophyton rautanei were chiefly composed of mono- and polyunsaturated fatty acids, whereas the oil of Ochna cerulata contained mainly monounsaturated together with saturated fatty acids. The first 2 oils, from the chemical point of view, are similar to tung oil, while Ochna cerulata oil resembles palm oil. The graph in the right upper corner illustrates that Ochna cerulata contained moderate amounts of both alpha and gamma tocopherol. The oil of Schizophyton rautanei had the highest level of gamma tocopherol. However, from the biological activity point of view, the gamma tocopherol has only 10% of activity compared to the alpha tocopherol. Furthermore, the oil of Parinaricura telefolia had negligible amounts of vitamin E. The last graph illustrates that the cakes remaining after the oil extractions are quite rich in some of the essential macro and micronutrients, including nitrogen, phosphorus, sulfur, natrium, potassium, calcium, and magnesium. The results indicate that all tested species have the potential of being introduced into the food industry as cooking oils. Especially Ochna cerulata deserves deeper research attention since it contains relatively large quantities of alpha tocopherol and has non-racid properties, and is preferred over the other oils by local communities. Due to their nutrient-dense cakes, the tested species also seem to be promising for being employed into animal feed. However, further studies are needed to reveal other technical properties, for example iodine, peroxide, or saponification values, and also verify the safety for human consumption. Thank you for your attention.
Thank you very much, Anna, for this very well prepared video.
Thank you.
It was very clear. I think you have one question in our chat from Mr. Singh.
Mm-hmm.
And the question is, are those genetic resources of underutilized plants, is there any value even to conserve it?
Mm-hmm.
What would you say?
Yeah, it was even you could have heard it from the video because I think we can't really rely on only maize, corn, and wheat, all these crops which are grown worldwide. So therefore, we really need to search for some other crops which can not really substitute those staples, but to really contribute to well-balanced diet of local populations. And also, I would say that one of the biggest advantages of the neglected, let's call it neglected species, is that they are really well adapted to local conditions. So they are not that vulnerable to droughts, to the effects of changing climate. So this is also another advantage, or why it is important to also conserve these type of species. And also, additionally, they are of big importance for the local communities because it can provide additional income. Also, in times of food scarcity, you still have some kind of species you can rely on to feed your family. So, yeah. And as I also said in the video, it's not only advantages for the developing countries, but also the developed countries can benefit from it, because if you take species such as baobab, which can be taken as a flagship of neglected crops, I would say, this is now introduced well to Europe as well. We have baobab yogurts and juices, and we can buy it here. And also, by buying it here, we are supporting the local communities in Africa, which are focusing on growing the species. So, yeah, I hope I expressed my idea at least a bit.
And I fully agree to your response. There's a lot of value in conserving those species and then actually developing pathways towards using them and enabling income generation for the local populations. I think also especially trees are playing in resilience in climate variable climate and change. So
Yeah,
it's definitely worth.
If we combine it with agroforestry and this sustainable agriculture practices, which
exactly.
Yeah.
Excellent. Yes, Mr. Wiele?
Yes, Anna, thanks for this talk. I would say truly neglected species. I have never heard of them.
Yes.
And my question would be, how did you select these species? Or what's their kind of priority list? Or did you assess it with participatory approaches to ask communities? Yeah.
Thank you very much for the question. Actually, this project is a development project, so it's not primarily scientific. So while working with the local communities, with the small-scale farmers as well as cooperatives, we realized that there are certain crops which have not been scientifically well described yet, or not at all in terms of Ochna cerulata. There are like 3 or maybe 2 articles published yet already. So based on the local preferences and what we saw in the field, we just tried to make some collection, do some sampling, and really see what's inside of these species. So we don't only work on these particular oil trees, we have some fruit trees as well. And right now we are just trying to somehow bridge the gap between the research and the development projects. So yeah, it wasn't really a methodology. It was just what we saw in the field.
Okay, great. Yes. Are there further questions? If not, I use the remaining 5 minutes for a final statement from each of you to the overall theme of this conference, namely food security and its resilience to global crisis. Did you understand? So I give you a few seconds to think about this. How does your study relate to food security, food and nutrition security, resilience to, and maybe everybody could talk like about 1 minute, and maybe because I put the spot on you, sorry for, but you are still We can't hear you right now.
Is this me?
Yes.
Okay.
Sorry. I think, yeah.
No, I would say, considering the number of species, the extraordinary number of species we are talking about, and the less information we have about most, it's of very high relevance actually for future and for conserving biodiversity. Yeah, so I would say this research of neglected and underutilized species should be much more in the focus of international and national agendas. And yeah, species can pretty much contribute to improved livelihoods at each country, at each levels, at each communities.
Thank you very much. So I would like to encourage you also maybe to write this down in the question and answer chat as response to Mr. Singh, who seems to. And now the next final statement, I would like to ask Claudio. Food and nutrition security and its resilience.
Yeah, I would say like a cocomia can be used for intercropping and also for smallholders. And like and the fruits are like so they have a versatile use, you know. So they're very good to get a better income for farmers.
Okay. Then the final statement of how does your study relate to food and nutrition resilience?
It's me? Okay. As my concern, as you all know, rice is the main staple food for half of the main food source for more than half of the global human population. So somehow the doing research related with rice, somehow, I mean, related to the food and nutrition security for the global human population, I guess.
Okay, yes, thank you very much. And now finally, a final statement.
Okay, thank you. I think we discussed this already during my poster presentation, but the neglected species are, I think, of high priority for the food security. And for the local communities, this is really crucial as a response to the changing climate, also to the price fluctuations on the local market. So yeah, focusing on this kind of species, the indigenous one, should be of high importance, also for us from the scientific point of view.
Okay, thank you. Final statements. Thank you for contributing a poster to this conference. It all worked out despite. And now the time is over. I wish you a good day, and maybe we'll see each other again in the next session. Thank you very much.
Thank you very much. Bye.
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 Bye.